Okay. All right. So I just did this whole thing and then I realized that I had forgotten to push the record button. So now I have to do it again, but that's okay. I love doing this stuff for you guys. All right. So what you guys are going to be making is a trifold pamphlet that has all of the stages of cellular respiration in all of their gory details. All right. So as we do this, today we're only going to do the overview. We're going to show glycolysis, and then we're going to show what happens to pyruvate if it stays in the cytoplasm without oxygen, and that would be fermentation, right? On another screencast, we'll go through all of the details of cellular respiration and oxidative phosphorylation, right? So this will be an assignment, you guys creating this pamphlet, and I'd be happy to laminate for you if you want it laminated afterwards. So this is going to be an assignment. I will post it in Whipple Hill. I'm going to go through all the drawings. Today we're doing, or on this screencast, I'm doing the overview and glycolysis and fermentation. All right, so let me share my screen. Okay, so I'm going to get a new page because I already did this, so I apologize. New page. All right, so we're just doing the front page of this trifold, which is all about the cellular respiration overview. So this is going to be cellular respiration overview, right? So in the overview, we have kind of two reciprocal processes. One, we have photosynthesis. And on the other side, we have cellular respiration, right? And they're kind of reciprocal processes. So the process of photosynthesis, and we can write out the equation here. I'm going to write that out in a little smaller. So photosynthesis, we're going to take energy from the sun. We are going to put in carbon dioxide, and we're going to put in water. And we are going to get out C6H12O6 and oxygen, right? And when we look at photosynthesis, we are going to see, so this is kind of the process of photosynthesis. Again, the inputs are going to be carbon dioxide and water. And we also obviously need some energy in the form of sunlight. And the outputs are going to be C6H12O6, so glucose, and oxygen. All right. So anytime I draw an arrow during a reaction, this blunt end are the reactants, and the pointed arrow are going to be the products, right? So photosynthesis converts carbon dioxide, light, and water into glucose and oxygen. On the other hand, cellular respiration, cellular respiration is going to do a reciprocal function. So let's write out the equation for cellular respiration. So for cellular respiration, we're going to see C6H12O6 plus oxygen is going to go to carbon dioxide and water and energy, right? And that energy is going to be obviously in the form of ATP, right? So if you write out the whole equation, and we can do this looking at a balanced equation, we have C6H12O6 plus 6 oxygen, and that's reciprocal reaction. So it goes both directions depending on which one you're talking about. You'll get carbon dioxide, water, and energy, right? And that energy can either be ATP in cellular respiration, or if you're going to photosynthesis, it's light energy. And I just want to remind you that the carbon, we're always going to keep our eyes on the carbon. So here, as glucose is being converted to CO2, that is going to be an oxidation reaction. And as oxygen is being converted to water, we're going to see that that is a reduction. So this is a kind of a redox reaction. Okay, so that is the overview. Now I'm turning to kind of the first page of my trifold, which is going to be all about glycolysis. So this is going to be glycolysis. And I'm going to put some key facts about glycolysis before we draw it out. So first and foremost, it is in the cytoplasm. It is in the cytoplasm of the cell, and it requires no oxygen, right? So you do not need oxygen to do that. Glycolysis is pretty special because it occurs in all living cells, right? From the bacteria in the thermal vent of an ocean all the way up to the skin cells on my fingers, right? So it's pretty rad. It occurs in all cells. It needs NADH to run, right? Or sorry, NAD plus to run. And we are going to see an output of 2 ATP, right? So for all of these reactions, we're going to put what comes into the reaction, what comes out of the reaction, all right? And in some of them, we're going to then look at the net inputs and outputs. So for glycolysis, we're going to look at what comes in, what comes out, and kind of the net, all right? So what comes into glycolysis, we're going to see that we're going to need, what comes in is 1 glucose, right? 1 C6H12O6. And then we're going to need 2 ATP, and we're going to also see 2 NAD plus. What comes out is that we're going to see that glucose now gets converted to 2-pyruvate. We're going to see that we're also going to make, or we're going to get 4 ATP out, and we're going to get 2 NADH out. So really the net overview in glycolysis is that we have 2-pyruvate, 2 ATP, and 2 NADH. All right, so glycolysis is unique. It happens in the cytoplasm, no oxygen necessary, but it occurs in these 2 different phases, all right? And I'm going to draw, whoops, not black here. I'm going to just draw a dotted line to represent these 2 different phases of glycolysis, all right? So the first phase is called the energy investment phase. So this is energy investment. All right, and so that energy investment phase, we're going to see that we're going to have to input energy. And then the second half of glycolysis is going to be the energy payoff phase, right? And that's when we're going to get energy out. So the energy investment, you're going to add 2 ATP, and the energy payoff, you're going to get 4 ATP, all right? And that's what we're going to see as we go through. So whenever I go through cellular respiration or photosynthesis, the key is to keep track of your carbons, all right? So I draw carbons as little circles. So glucose has 6 carbons, right? And if you zoom in, you can label these 1, 2, 3, 4, 5, 6 little carbons, right? So the energy investment phase here, we're going to go through. It's going to be several different enzymes that convert this sugar into different things. So we're going to have 1 enzyme, 2 enzymes, and 3 enzymes. And those 3 enzymes, we don't have to know exactly what they do, but your book has lots of gory details. But what they're going to do there is that we're going to have 1, 2, we're going to see that glucose, actually, I'm going to move that over a little bit. So we're going to see that those 6 carbons, 1, 2, 3, 4, 5, 6, those carbons are going to be 2 of them, the first and the 6th carbon, are going to become phosphorylated. And again, a phosphorylation just means that you're putting a phosphate group onto either carbon number 1, carbon number 6. Here we're doing both, all right? And what we see as we do that, we're going to see that we have our investment, we're going to have ATP is going to be converted to ADP, right? So you see 1 phosphate, and that is where that phosphate is going to come from. We're going to see another phosphate come from the hydrolysis of ATP down there, right? So we have 3 enzymes, but all you need to know is that carbon becomes phosphorylated on both ends, right? And then what's really cool is now we get to the glycolysis. Can you close the door, Tove? The glycolysis, which is glycolysis, right? So we're going to see that this molecule is going to get cut down the middle, right? Glycolysis. So we're going to see quickly that, whoops, let me go back to black here, is that, oh my gosh, why is that doing that? Sorry. Is that we're going to see this molecule now be split into 2 3-carbon sugars. So we're going to see 1, 2, 3, and 1, 2, 3, right? And that is still going to be phosphorylated, right? So now we have 1, 2, 3 carbons and 1, 2, 3 carbons. All right, so that is the energy investment phase. So what we've done is phosphorylated glucose on both sides and cut it in half. All right, so the next part is a whole lot of details, which you don't have to know very much of, but I'm going to tell you there's 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Yes, there's going to be 5 enzymes. 1, 2, 3, 4, 5. That do this to both of those 3-carbon sugars. And what you're going to end up with are 2 pyruvate molecules. In a pyruvate, you don't have to know the details, but a pyruvate represents a 3-carbon sugar, right? So you get 2 pyruvates out, right? As you're going through all those enzymes, I told you there's 5 enzymes, there's some things that happen, all right? So first off is that we're gonna see that NAD+, which I told you that we're gonna need, is gonna make NADH, right? And it's gonna do that on both of these, right? So what that's gonna end up with, so what that's gonna end up with is that we're gonna see 2 NAD from that first reaction, right? 1 from each side. The second enzyme, we're gonna see that we're gonna create ATP. So remember, we're doing energy payoff here. So we're gonna get 2 ATP there. And then at the end here, we're gonna see that we get another 2 ATP. So ADP is being converted to ATP here. ADP, and this I just want to make it real clear, ATP, all right? So in this process of glycolysis, we had energy investment in the form of 2 ATP in at the top. In the energy payoff phase, we get 4 ATP out, and we get 2 NADH out, right? So you can go back to the net inputs and outputs and make sure that everything makes sense, right? So that's glycolysis. And we know, again, that if we look at these pyruvates, they can have 2 different paths depending on if there's with oxygen or without oxygen, right? So if we have lots of oxygen, that's going to go to our mitochondria. If there is no oxygen present, then we're going to stay in the cytoplasm, all right? And that's what we're going to talk about next. Okay, so this one, we are going to talk about fermentation. Let me pull up mine. All right, so here we are talking about fermentation. Process of fermentation. Just make sure I have everything. Okay, and just like we did for glycolysis, we're going to write a couple facts. So this is going to happen in the cytoplasm. This no oxygen required. It has different reactions in different organisms, right? And we'll talk about those 2 kinds of fermentation. And the whole purpose of fermentation is to regenerate NAD+ So glycolysis can continue. And that's really important so that we can make some ATP, even in the absence of oxygen, right? So remember our input here are our 1, 2, 3 carbon molecule, right? So that is pyruvate, our 3 carbon molecule that we got from, which is the product of glycolysis. And depending on what organism you are, you can do 2 different things. So we're going to first, on this side, talk about lactic acid fermentation. And that happens in most animal cells. I gotta look it up. I think it happens in other things too, in plant cells, but I'll look that up. And then the other kind of fermentation is specifically, or is called alcoholic fermentation, right? And that one is very specific to yeast cells. But the reason it's so important is that we use a lot of products in our society based on this alcoholic fermentation, right? So our beer and wine and bread and a lot of things depend on that. So for this process, let's talk about lactic acid fermentation first. So pyruvate in the cytoplasm is converted to lactic acid, right? So we have pyruvate and it gets converted to lactic acid. And in the process, of that, NADH turns into NAD+, right? And that's an oxidation. Remember, you're going to remove the H. So that's an oxidation. Oof, sorry. And that happened, and that's it. And so when you are running sprints, you have a reduced amount of oxygen in those muscle cells because you've used most of that oxygen up, and therefore your cells still need to make energy. And they need to do that through glycolysis. So cells go through fermentation to regenerate NAD+, so that you can continue to make some ATP through glycolysis. All right, if you are a yeast cell, which none of you are, thank goodness. So if you are a yeast cell, you go through alcoholic fermentation and you make ethanol, right? You make ethanol through this fermentation. And you get the same thing, so you have to put in the same, you're going to put NADH in, and again, you are going to get NAD+ out. And again, the whole purpose of this fermentation is to regenerate your NAD+ so that you can continue to do glycolysis and get a little energy. The other thing that's really cool about alcoholic fermentation is that you get CO2 out, right? And that is why that is important for making bread rise, right? The bubbles in that gas help bread rise, right? Okay, so let's talk about the ins and the outs. So I should have done this at the top, and I apologize, but I did not. So we're going to do the in and the out and the net. All right, so in, we're going to see pyruvate. Pyruvate in, and we're also going to see NADH get put in. Out, we are going to see NAD+ is going to be one of the main outputs. And then depending, you can either get lactic acid if you're an animal cell, or you can get carbon dioxide and ethanol if you're a yeast cell. And so the net is really NAD+, and that is to help glycolysis continue so that you can continue to make some ATP. All right, so we're going to stop there. Next screencast will be all the drawing of kind of the aerobic respiration part.